Alright, let's break down the enzymes and molecules that power your cells, cellular respiration style. I'm going to walk you step by step through the main players, so by the end you'll actually know what they do. NADH, NAD+, FAD+, FADH2, and acetyl-CoA. Picture your cell as a tiny factory. Cellular respiration is its energy-making assembly line. The process has three big stages: glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis splits glucose into two pyruvate molecules in the cytoplasm. The Krebs cycle finishes breaking down carbon and collects high-energy electrons. The electron transport chain turns those electrons into a proton gradient to make ATP. First, hexokinase adds a phosphate to glucose. This traps it in the cell and primes it. Hexokinase uses ATP to form glucose-6-phosphate, no going back out of the cell. Phosphofructokinase is the pace setter. It commits the sugar by making fructose 1,6-bisphosphate. The 6-carbon sugar splits into two 3-carbon molecules that continue down the line. During this, NAD+ steps in to capture high-energy electrons. NAD+ is like an empty shuttle bus looking for passengers, electrons and hydrogens. It grabs electrons and hydrogen from the pathway, turning into NADH, a full bus loaded with energy. So, NAD+ becomes NADH and carries those electrons forward. NADH's job is transport, delivering its electrons to the next stage for ATP production. Glycolysis nets a small ATP profit and makes NADH, then sends pyruvate to the mitochondria. Next, pyruvate moves into the mitochondrion to be processed for the Krebs cycle. But first, acetyl-CoA shows up, the VIP pass that gets carbon into the cycle. The enzyme pyruvate dehydrogenase removes CO2, captures electrons as NADH, and attaches CoA, forming acetyl-CoA. Without this step, the cycle doesn't even start. Acetyl-CoA is the ticket onto the ride. Inside the Krebs cycle, enzymes break down acetyl-CoA and harvest electrons. Citrate synthase begins the cycle by forming citrate, starting the carbon journey. Aconitase rearranges citrate into isocitrate to set up the next reactions. Isocitrate dehydrogenase removes CO2 and reduces NAD+ to NADH, key control point. Alpha-ketoglutarate dehydrogenase repeats the pattern. CO2 out, NADH made, CoA attaches. Succinyl-CoA synthetase makes ATP or GTP directly. Bonus payout. Here's where FAD+ comes in, another electron carrier like NAD+, but bound at complex 2. FAD+ accepts electrons and hydrogens from succinate, turning into FADH2. By cycle's end, you've built a stack of NADH and FADH2, full shuttles ready to deliver. NADH unloads at complex Y. FADH2 unloads at complex 2. Slightly different energy yields. Their final destination? The electron transport chain, a high-tech conveyor belt in the mitochondria. As electrons flow, membrane enzymes pump protons out, building a gradient, like charging a battery. The final enzyme, ATP synthase, lets protons flow back to power ATP production, your cell's energy currency. Oxygen is the terminal electron acceptor, forming water and keeping the chain moving. So, to recap, NAD+ and FAD+ are empty shuttles. NADH and FADH2 are full shuttles carrying electrons. Acetyl-CoA gets the Krebs cycle rolling, and enzymes turn food into ATP so you can move and even scroll TikTok.